Okay, good afternoon, everybody. It's my pleasure to introduce our speaker today, who's Niels Okkels from Aarhus University in Denmark. And well, I know Niels I got to know him about 4 years ago when he came as a PhD student to do a short-term internship in my lab in Seville to learn about neuroimaging. And this brief interaction has turned into a very fruitful collaboration over the last years. And I'm really happy to have you here today, finally. And yeah, today, Niels is a junior group leader at Aarhus University and PI of several research grants related to dementia with Lewy bodies, Parkinson's, and also Alzheimer's disease. I'm really looking forward to hear about them.
Well, thank you for that wonderful introduction. I must say I'm very overwhelmed to be here. Not at least because it's at least 20 degrees warmer here compared to back home. And also, I just went for a wonderful lunch with some traditional paella and whatnot. So I'm a bit overwhelmed here. Yes, I'm associate professor of dementia research, and I'm also a medical doctor enrolled in the neurology specialization program. And my focus is dementia with Lewy bodies, but also in the recent years, I have taken up Alzheimer's disease. And all in all, my hope is that my research can contribute to a more timely and accurate detection of these neurodegenerative disorders. At the recent ADPD conference in Copenhagen, I actually attended a few of your talks here from this group, and I just wanted to congratulate you on the work that you are doing here at the CIEN. I think it's quite fantastic. Yeah. I recently took the first step to establish my own group as main supervisor for 3 PhD students and a number of pre-graduates. My work is primarily funded by an early career clinician scientist grant from the Lundbeck Foundation and a clinical emerging investigator grant from the Novo Nordisk Foundation. And if there are any clinician scientists in the audience here today, please let me know if you consider applying for one of these grants, and I should be happy to review your application before you submit it. The main focus of my research and the topic for the 2 talks today are in vivo biomarkers of Lewy body disease and Alzheimer's disease, specifically blood-based biomarkers, which will be the subject of the first talk, and imaging-based biomarkers, which will be the subject of the second talk. So in this first talk, I will present some preliminary results from analysis of more than 1,000 longitudinal preclinical plasma samples collected over a 15-year period from Danish blood donors who later developed Alzheimer's dementia. We also have samples from donors that developed Lewy body dementia, but those analyses are not ready to be presented yet. The project is based on Danish blood donors. While blood donation systems exist, I assume also here in Spain, Denmark is unique in 2 ways. Donors provide broad consent for research, and we can link their data to national hospital registers. This gives us a unique source population of about 170,000 individuals with over 1 million person-years of follow-up and more than 2 million archived plasma samples. And from this cohort, we identified a subset of donors who were later diagnosed with dementia and obtained access to their previously donated plasma samples for biomarker analysis related to Alzheimer's disease. The project addresses 3 critical gaps in the literature. First, we have access to samples collected over 15 years before diagnosis and soon up to 20 years, allowing us to map very long preclinical trajectories. Second, we have multiple repeated samples from the same individuals, enabling longitudinal with in-person analysis. And third, many of these donors are still alive, which means that we can invite them for detailed clinical assessments, including advanced PET imaging and MRI imaging. So this is an overview of the study design. In the top left corner, you see a typical blood donor who donates repeatedly over several years. At each donation, 1 milliliter of plasma is stored for research. Then, using register linkage, we identified donors who later developed dementia and retrieved their stored plasma samples to analyze for p-tau217, NFL, and GFAP on the Simoa platform. The median age of the total Danish blood donor cohort is 37 years. So even though there are 170,000 Danish blood donors, until now, only 258 have been diagnosed with Alzheimer's dementia. Overall, their mean age is 69 years at time of diagnosis, and there's an equal sex distribution. The incidence curve here shows a supralinear increase, and we expect to identify at least another 500 individuals over the next 5 years. Because donors can give multiple blood samples each year, we included only samples collected at least 1 year apart. Overall, there's a median of 5 samples per donor, totaling approximately 1,400 samples. Today, I will present the preliminary analysis from 165 cases and a similar number of matched controls, comprising around 1,600 serial samples. The analyses are ongoing and have been developed in close collaboration with my PhD student Celia, who has done much of the foundational work. This figure shows p-tau217 concentrations over a 15-year period in individuals who were later diagnosed with Alzheimer's dementia. The red line represents the median with interquartile range, and the dashed lines indicate cutoffs at 0.4 and 0.63 picograms per milliliter. Although these thresholds were not developed for preclinical samples, it is notable that concentrations cross the lower cutoff more than 10 years before diagnosis, and then increase gradually over time before crossing the higher threshold. Here we have added matched controls. Because the Danish blood donor cohort is so large, we were able to perform very strict matching. So each case is matched not only on age and sex, but also on geographical region, and importantly, on the number and timing of blood donations. This means that cases and controls have essentially identical sampling patterns over time, which is quite unusual in this type of study. What stands out is that p-tau217 levels in cases are consistently elevated across the entire 15-year period with minimal overlap with controls. In this figure, cases are stratified by sex. Blue represents men and red represents women. Although formal statistical testing is still ongoing, the trajectories appear remarkably similar between men and women. In fact, so far our data indicate that there's no difference between the two sexes. Here we show box plots for the first 8 years before diagnosis. These findings are broadly consistent with previous studies covering the first 6 to 7 years prior to diagnosis. However, an important point to consider is that many donors stop donating blood around the age of 68. This means that for individuals diagnosed later in life, for example, in their 70s or 80s, there can be a substantial gap between their last blood donation and the time of diagnosis. And we're still trying to investigate how this influence the box plots, particularly in the years immediately preceding diagnosis. This figure extends the analysis to 9 to 15 years before diagnosis. There are relatively few samples at the earliest time points. So those estimates should be interpreted with caution. That said, the data suggests that p-tau217 is already elevated at least 15 years before diagnosis. This is supported by ROC analysis showing AUC values above 0.8 across the entire preclinical period. However, it is important to note that this reflects discrimination between Alzheimer's diagnosis versus no diagnosis. It does not account for other dementia types. Using linear mixed effects models, we find that these preclinical elevations may extend even further back in time, potentially beyond 15 years. We are currently exploring whether our data can support this. Among controls, we observed a bimodal distribution of p-tau217. One hypothesis is that a subset of controls may be in a preclinical disease stage. However, when examining individual trajectories, these values remain stable over time, suggesting a different explanation. Within a multi-center framework, my PhD student Søren Okkels will now invite donors for detailed clinical assessments, including neuropsychological testing, advanced imaging, and collection of fresh samples. This will allow us to evaluate the clinical relevance of preclinical biomarker changes. Søren's work is co-supervised by Pascal Sanchez, and I hope that Søren may one day come to visit your center here and discuss his work with you for a short research stay in a year or two from now. There are several limitations to consider in this work. First, a register-based dementia diagnosis have an estimated error rate of about 20%. We will address this through medical record validation and clinical follow-up. Second, we know that comorbidities may influence biomarker levels, and we will use national registers to assess conditions such as chronic kidney disease. Third, blood donors are generally healthier than the background population, the so-called healthy donor bias. However, it is important to note that donors are not excluded based on key Alzheimer's risk factors such as hypertension, diabetes, dyslipidemia, or smoking. This means that despite the healthy donor bias, the cohort still captures many of the most relevant vascular and lifestyle risk factors in Alzheimer's. Also, it is important to mention that while I have referred to these samples as preclinical, we do not know when symptoms began, so pre-diagnostic may be a more accurate word. And finally, validation studies conducted by our collaborator Inge Verberg in Amsterdam suggests that long-term storage at -20 degrees Celsius does not significantly affect biomarker measurements. So to conclude this first talk, I would encourage you to consider this study design in a broader perspective. From the discussions I had today, for example, I learned that you are also here interested in frontotemporal dementia. Using archived donor plasma to study pre-symptomatic disease could be applied to many neurological disorders with long prodromal stages. So in this data set, we also have, as I mentioned, donors diagnosed with dementia with Lewy bodies, and we will present the preliminary results at the Next Gen DLB webinar on May 7. My colleague Per Borhammer is currently collecting data on donors diagnosed with Parkinson's disease, other conditions such as REM sleep behavior disorder, atypical Parkinsonian disorders, ALS, and FTD may not yet be ready for this type of approach, but are likely to become so in the very near future. Yep, so this was the end of my first presentation. Thank you for your attention.
Niels, should we have some Q&A right now, right
Yes.
away? I think that maybe.
Yes,
right.
let's do that.
Really nice. I would say really nice design. I wonder, it's more of a curious question because in the beginning you said like with the SIMWA you also measure GFAP.
Yeah.
And I wonder, you didn't show the results in the presentation, so I was wondering, do you see also these differences in very preclinical phases between GFAP? Can't you disclose?
I don't know yet, unfortunately. Yeah, I was about to say that we will present those results at the AAIC, but I don't think they will be ready yet for that. But they are working on GFAP and NFL in the lab right now. But yeah.
So I wonder about the longitudinal differences because you said like for each donor you may have a serial sample. So I wonder like the change between, for example, the first two samples is something to look like the gap. The change in biomarker may also be more indicative in the first years than maybe the whole trajectory.
Yeah. I very much agree. That's on the very top of the to-do list because I very much agree with you that to look at the dynamics between serial samples is what I think will inform the clinical work the most. Imagine that you have
you had this graph. It was only a little figure, but with the no, a bit earlier with the healthy controls that this one.
Yeah.
So the healthy controls that have similar intermediate values, but they don't progress. So maybe measuring the change between visits can be more informative than the baseline value itself.
Yeah, I agree completely. Yeah.
The cohort of blood donor that you have, do you think that it is representative of the overall population? For instance, education, socioeconomic status, and so on, is similar. It would be considered as representative.
Yes, I think that's a wonderful point, and I think you are completely right, namely that on average, blood donors are healthier and probably also better educated than the background population. And what we are doing right now is that we are collecting data from the entire nation on dementia diagnosis so that we can compare our group of blood donors with a dementia diagnosis to a typical Danish person with a dementia diagnosis to understand how big is the difference in lifestyle and in education and so on, to understand how we can generalize these results, if we can generalize these results to the general population. So I think it's a wonderful point, and we are working to explore that. Yeah.
I was thinking when you said this unique possibility that you have to perfectly match the controls.
Yeah.
I was thinking about a bit exotic idea that maybe you can match for the ApoE genotype and comparing cases and controls with exactly the same ApoE genotype, you can extract what is the difference, because ApoE is such a powerful drive that it would be very nice design to compare these two population extracting ApoE.
Yeah.
So you would be able, for example, you do proteomics, what makes difference individuals with the same genetic structure, at least in terms of ApoE, of course, not on the other genes, to see what are difference between these two populations. I think, of course, that will be a different set of controls.
Yeah.
But if you have the budget, I think that would be a unique thing to do.
Yeah. Yeah, I think that's a great point, and I wish that I had thought about that before we selected the controls, because we would in theory have been able to do that. Yeah. I don't know if we can do it. I don't know if maybe, but what we are working on right now is to analyze the ApoE genotype on those that we have selected already. Hopefully they will match.
Just a quick question regarding the problem you mentioned, that you don't really know if this is how much of it is clinical, and hence the term pre-diagnostic.
Yeah.
What about the do you have any detailed clinical information on these MMSE scores to know the degree of dementia severity at time of diagnosis? If these are, I don't know, primary care diagnosis, then they are probably relatively late, so at a relatively advanced dementia stage, at least in terms of research. So then you could exclude, at least, for example, like the first or the last 3 to 4 years before the registered diagnosis.
So yes, this is a very heterogeneous group. We included everyone with a Alzheimer-like diagnosis. No diagnosis in Denmark are made in primary care. All are made mostly by a neurologist in hospitals. So in general, the quality of the diagnostic workup is relatively good. What we have permission to do now is to go into everyone's medical record, electronic medical record, and we will do that now to firstly to validate the diagnosis and exclude those that had a wrong diagnosis or a diagnosis based on a very poor, insufficient workup, but also to collect some basic information about the MMSE or MOCA or ACE or yeah. But I think the best clinical information that we will have here is what we will collect ourselves by inviting the donors to come to the center. Everything will be standardized there and very detailed. Yeah.
I wonder if the routine neurological workup in Denmark includes the plasma biomarkers. So because maybe if you go into the medical records to see like how the diagnosis was performed, and you eventually find a new biomarker level, maybe you will have like the pinpoint exact level at the time of the diagnosis that you can compare to the previous values. I don't know if I'm explaining this.
Okay, I think that's very interesting.
For this, you would need to have the routinely.
My fast reply here will be no, we cannot do that because plasma biomarkers are not used in the routine clinic. But I like your idea, so maybe we can elaborate on it maybe afterwards. Yeah.
So I don't know if there are no further questions. Maybe we should move on to the next part.
All right. Okay, so now I would like to share my second presentation, which focuses on imaging biomarkers, specifically cholinergic PET using the tracer FEOBV. Much of the work that you will see in this presentation was made in collaboration with my colleague Michel and also Miguel. So in the next 20 minutes, I will introduce some important points about dementia with Lewy bodies, DLB for short, and the cholinergic system, followed by the main methods used in our studies, namely cholinergic PET using FEOBV. The main part of this presentation will focus on our findings pertaining to the brain, but also the peripheral organs. As many of you maybe are familiar with, dementia with Lewy bodies is clinically characterized by dementia plus core features of fluctuations, hallucinations, REM sleep behavior disorder, and Parkinsonism. But what many of you maybe don't think about is that dementia with Lewy bodies is a multi-organ disease, and pathological alpha-synuclein appears abundant in not only the brain and spinal cord, but also in the peripheral nervous system, the gastrointestinal tract, the endocrine and exocrine organs. As such, Lewy body pathology and neurodegeneration affects cholinergic neurons across the central and peripheral nervous system. And cholinergic dysfunction is implicated in several key features of Lewy body disease, including, of course, cognitive dysfunction, but also impairments in gait and balance, hallucinations, hyposmia, sleep disturbances, and autonomic dysfunction. Cholinergic neurons are defined by their ability to secrete acetylcholine. Some, but not all, of these classes of cholinergic neurons seem vulnerable to pathology and degeneration in Lewy body disease. The findings presented today focus on cholinergic projecting neurons in the brain and parasympathetic neurons in the thorax and abdomen. In brief, cholinergic projecting neurons in the brain affect how other neurons react to stimuli. For example, cholinergic neurons may decrease response to stimuli that do not require immediate action or potentiate behaviors that are adaptive. The general function of the parasympathetic neurons is to stimulate activities that occur when the body is at rest, including digestion, urination, defecation, salivation, lacrimation, and sexual arousal. The cholinergic system is very important to cognitive function, and this is known because anticholinergic medication affects cognition in healthy individuals, cholinergic neurons degenerate in dementia, and procholinergic medication can improve cognition in patients with dementia. Throughout the central nervous system, there are various clusters of cholinergic nuclei, and several of these nuclei play important roles in cholinergic neurodegeneration. In the context of this presentation, it is crucial to highlight that the dorsal motor nucleus of the vagus, here in the medulla oblongata, projects its parasympathetic fibers to innervate visceral organs, and that parasympathetic fibers are cholinergic because they transmit acetylcholine. Collectively, the majority of these cholinergic cell groups are characterized by being significantly affected by pathology and neurodegeneration in Lewy body disease. Another important point to emphasize is the differences in binding targets between FeOBV and other cholinergic tracers commonly used in cholinergic research. While FeOBV specifically binds to the vesicular acetylcholine transporter, which is a very specific marker of cholinergic terminals, the bulk of cholinergic research has been performed with cholinergic tracers targeting less specific molecules, typically acetylcholine esterase. FeOBV has undergone validation for its use in both the brain and peripheral organs. In the brain, the distribution aligns with histological staining and expression of cholinergic genes. In peripheral organs, FeOBV demonstrates binding in organs that are known to exhibit cholinergic activity. In our studies, both those conducted in the previous years and ongoing studies, we conduct brain FeOBV PET on patients with DLB, Parkinson's disease, Alzheimer's disease, and healthy controls. In addition, we conduct whole body FeOBV PET on a subset of these patients. In addition to the PET scans, we also perform a comprehensive battery of clinical and cognitive assessments. I will not go through how we analyze the brain data, but this will probably be new to you, so I'll just mention it briefly. The calculation of PET uptake in peripheral organs is performed manually by outlining a region of interest. So this figure represents A, the liver, B, the kidney, C, the heart, and D, the pancreas. Now I will share our main findings related to the peripheral organs, and following that, the findings related to the brain. In our studies, we made an interesting observation regarding the colon. We found a significant decrease in uptake of PET tracer among patients with DLB compared to healthy controls. And we believe that a likely explanation for this is parasympathetic denervation. A previous study demonstrated that colonic, yes, sorry, this is SUV. Yeah. Yes, so we believe this is a signal of parasympathetic denervation because a previous study found a reduced uptake in patients who had undergone vagotomy, which is a surgical disruption of the vagus nerve delivering parasympathetic innervation to the colon. We also observed a decreased cholinergic uptake in the pancreas in DLB patients compared to controls. The pancreas is an organ situated below the stomach and plays a crucial role in digestion and also regulation of blood sugar levels. However, the exact explanation for decreased cholinergic uptake in the pancreas is not entirely clear. For example, alpha cells within the pancreas also secrete acetylcholine to stimulate beta cells, which are responsible for insulin secretion. Additionally, stellate cells also within the pancreas secrete acetylcholine to stimulate exocrine function. So considering these factors, it is evident that the cholinergic integrity of the pancreas may involve some non-neuronal cholinergic uptake mechanisms. Nevertheless, the significance of our findings in the colon and pancreas is underscored by the absence of group differences in organs that have little or no parasympathetic innervation. Now that I have presented our findings pertaining to peripheral organs, I would like to share our most important findings regarding the brain. This figure represents axial slices showing differences in cholinergic uptake among healthy controls, patients with Parkinson's disease, and DLB. And by comparing the images, you may appreciate a lower uptake of cholinergic tracer, particularly in cortical regions. In addition to the cortical regions, several other brain areas exhibit noteworthy differences in cholinergic uptake, and these include the olfactory cortex, the lateral geniculate nucleus, the cingulum, the amygdala, and the hippocampus. In the global neocortex, we found a severe and widespread reduction in DLB patients compared to PD patients. The figure to your right compares results from published cholinergic PET studies investigating neocortical cholinergic activity. And what you can notice here is that almost all studies in PD report global neocortical reductions of less than 20%, whereas almost all studies in DLB report reductions that are greater than 20%. Moreover, when comparing DLB patients to PD patients, we observed minimal or no differences in certain regions, for example, in occipital regions, whereas other regions, typically limbic regions, here exemplified by the olfactory cortex, exhibited very large differences. As you are probably well aware, since you work with Michel, the cortical and limbic regions of the brain receive cholinergic innervation from cell bodies located in the basal forebrain. And to investigate potential atrophy of this region, I traveled to Michel and Miguel in Seville to learn how to use MRI to assess the volume of the basal forebrain as a proxy measure. Our findings revealed decreased volumes of both the anterior and posterior regions of the basal forebrain in patients with DLB. And interestingly, we observed that the degree of atrophy was more prominent in posterior regions compared to anterior regions, which supports that the pathological process in DLB may progress through the basal forebrain in a posterior to anterior direction. Furthermore, patients with PD exhibited significant reductions in cholinergic terminals, but preserved volumes of the basal forebrain. And this discrepancy may suggest that cholinergic terminal dysfunction may occur prior to atrophy of cell bodies in the basal forebrain. We also found that cholinergic terminal binding in limbic and cortical regions correlated quantitatively and spatially with subregional basal forebrain atrophy and DLB. This suggests that pathological processes in the anterior basal forebrain and corresponding limbic cholinergic dysfunction is important for the development of dementia. In this figure, the red region marks the pedunculopontine nucleus and laterodorsal tegmental nuclei, which are together form a large group of cholinergic cell bodies on the junction between the pons and the mesencephalon. The green region marks the approximate location of the dorsal motor nucleus of the vagus. In our PET studies of the brainstem, we identified a distinct signal corresponding to these two cholinergic cell clusters. And interestingly, PET uptake in both these regions correlated with autonomic symptoms. This suggests that the integrity of cholinergic function in the brainstem may contribute to the manifestations of autonomic symptoms in this patient group. Additionally, our findings revealed a correlation between orthostatic hypotension, which is a common manifestation of autonomic dysfunction, and cholinergic terminal loss in a specific region corresponding to the dorsal motor nucleus of the vagus. These results suggest that loss of cholinergic terminals in the dorsal motor nucleus of the vagus may play a role in the development of orthostatic hypotension and contribute to autonomic dysfunction in patients with DLB. Alzheimer co-pathology is frequent in patients with Lewy body dementia, and therefore we speculated that the observed neocortical lobar pattern in Lewy body dementia may to a certain extent be a combination of that seen in Alzheimer's disease dementia and more pure forms of Lewy body disease. Studies in Parkinson's disease suggest that cholinergic denervation is most pronounced in posterior cortical regions compared to anterior regions. Studies in Lewy body dementia report a more pronounced but similar pattern extending to more anterior regions. In Alzheimer's disease dementia, the temporal region is generally the most affected region, followed by the parietal, occipital, and frontal lobe. Across studies, no individual cortical lobe in either Parkinson's disease or Alzheimer's disease is more affected compared to Lewy body dementia. Also, it seems to be a general finding that the frontal region is the least affected region across all groups. So, our data may support that both Lewy pathology and Alzheimer pathology contribute to the cortical cholinergic denervation observed in Lewy body patients characterized by frequent Alzheimer co-pathology. So, this line of studies suggested that Alzheimer co-pathology may significantly affect the pattern and severity of neurodegeneration in Lewy body disease. To study this further, we last year started a new series of studies which are being conducted by my PhD student Emil Breinholt Sørensen and in collaboration with Michel Grote. We are currently recruiting patients with varying degrees of biological Alzheimer's and Lewy body disease. Specifically, we characterize all patients at baseline on beta amyloid, tau, and alpha-synuclein, the latter being analyzed on the seed amplification assay. This will allow us to transcend the clinical entities to understand the biological impact of these pathologies on neurodegeneration, and in particular, the loss of cholinergic neurons. Another dimension of these studies is that we aim to understand the impact of Alzheimer co-pathology over a 7-year follow-up period, and hopefully, as one of few prospective studies, demonstrate that DLB patients with Alzheimer co-pathology have a more severe trajectory towards faster decline and death, providing an argument for enrolling patients with DLB and a certain amount of Alzheimer co-pathology in trials of amyloid targeting medication. The first preliminary results from these ongoing studies will be presented at this year's AAIC in London. And to finalize this talk, I would like to share one very small, but I think very surprising finding. For the studies so far, we have recruited what we believe to be 12 healthy elderly controls. However, the results from the seed amplification assay of alpha-synuclein came back positive in 4 of these participants. We initially interpreted that finding as false positive, but we then discovered that these 4 individuals had lower scores on measures of olfaction, while all other measures being similar between the 2 groups. So, as you may know, hyposmia is one of the strongest predictors of impeding Lewy body disease. So we then looked at the cholinergic PET images, and there we found a small but very characteristic and highly significant pattern of lower cholinergic uptake in posterior cortical regions. So we believe that what you see here may be the first cholinergic images of very prodromal, almost pre-symptomatic group of individuals with incidental Lewy body disease. And we are currently working to understand if this signal is robust enough to present in a paper. And what I will do first thing when I come home is to do polysomnographies to make sure that these are not just patients with REM sleep behavior disorder, although the RBDSQ questionnaire indicate that they are not. Thank you for your attention.
Wonderful presentation, Niels. I'm sure there are a lot of questions here.
Presentation? Will Emil have
Yes, it's an oral presentation in a session with the title "An Update on Imaging and Fluid Biomarkers in Lewy Body Dementia," chaired by Ramona Ryle from UCL, I think.
So maybe I think since the slide is already up, we should stay here for a while. And talking about the proportion, there are already quite some SAA studies from cohorts, Biofinder cohort or other big cohorts out. Do you know the proportion that has been reported in healthy controls in these other cohorts? Do you think you were just lucky to have like a 25, 30%?
Yeah, of course, this proportion is a lot higher, a lot larger than would be expected based on the Lund data. Overall, I would say that in people between the ages of 50 to 80, you would find around 8% who are who should be positive on the seed amplification assay for alpha-synuclein, and in our case, it's more like 30%. But what we also know is that if you select those individuals who score low on the olfaction test, the proportion is known to be a lot higher, maybe even 30%. The other thing I want to say is that based on the experience from our previous studies, when we identify healthy controls, we announce in the local newspapers. And we generally have a relatively large proportion that score an unexpectedly large proportion that perform poorly on the neuropsychological tests. So I think that our way of recruiting healthy controls introduces a bias because people who may consciously or unconsciously know that or feel that something is coming, they volunteer for these kind of studies. So we have a selection bias in the way we recruit healthy controls. Yeah, I hope I made that point clear. Yeah.
Thank you very much for your presentations. They were really interesting. And for me, it was very surprising the results of the whole body PET and how can they help to differentiate both types of dementia. And I was thinking about how these changes can happen in time, like if the changes, for example, in the pancreas or in the colon happen first, or the changes in the brain are first, or are both together?
Yeah. So I think that's a wonderful question, and of course, the short answer is that we don't know. The pathology, if we believe it's a prion-like pathology, it can likely travel in both directions, meaning either from the central nervous system towards the peripheral or the reverse. But what we do know about the prodromal period of patients with Lewy body dementia is that they often report, for example, constipation or other signs of autonomic dysfunction in the prodromal period, 10, 15 years before they start to develop cognitive symptoms. So that could indicate that the pathologies, at least in a large proportion of patients, start somewhere in the peripheral nervous system and then later starts to involve the brain. Yeah.
A very short question related to what Marina was saying. Are you aware of any problem related to diabetes or malfunction of the endocrine pancreas in patients with alpha-synuclein? Is there any?
We know that they have a slightly higher prevalence of diabetes, but not very much. So yeah, of course, I looked into that literature after seeing this finding, but to be honest, I don't think that there's a strong signal that this affects your risk of diabetes. No.
I think this is an interesting topic. That said, I think we know from a long time that diseases that occurs with alpha-synuclein affect other parts of the body. So MIVG expect has been a test for Parkinson for the last 20 years, and it's very well clinically established. That said, one limitation of a brain PET is that we only look in the brain, and now with the whole total body scans, there are a lot of projects. Actually, there's a project in the Netherlands to do whole body amyloid PET in Alzheimer's patients just to see if any accumulation will arise anywhere. That being said, when I asked about the SUV, which was the scale, it was because the values in the healthy controls were around 1. So this is in basic PET theory. This means like you are testing for a very specific uptake target. So a value of 1 means that there is no specific uptake in that region. So if you have 0 of these receptors in the whole body, this SUV of the whole body would be 1. So a value of 1 means there is no specific uptake. So going from 1 to 0.8 actually probably wouldn't mean like you are losing the receptors because the value is already 1.
Yeah, I think it's a very qualified comment, and I have to admit that it's been a year or two since I studied the theory. So, but I can get back to you with a good reply for that one. Yeah.
You take into account the medication, the cholinesterase inhibitors. Does it affect the images or?
Yeah, that's also a very good question. And yes, and the answer is this. When you look at evidence from mice and rodents that were scanned using cholinergic PET tracers targeting the vesicular acetylcholine transporter. When you looked at mice and rodents that were on, for example, donepezil or without, you saw no difference. So it should not affect our images, no. Yeah.
Hi, thank you for your presentation. I have more of a methodological question regarding the whole body PET approach that you're using. Because traditionally, in a lot of brain PET modalities, we use a reference region for quantification, such as the cerebellum. In this case, what do you use? Because I can imagine that it's quite difficult maybe to isolate a specific reference region that's devoid of that tracer uptake. Like, do you use like a quantification approach for each organ separately, or how does it work?
Yeah, I think that's a great question, and I wish I could answer it straight away, but I will have to look up on the methods and get back to you with that one.
So I have a question regarding the latest study that you presented with the mixed pathology using AD and alpha-synuclein biomarkers. Next slide. No. Yeah, this
one. Okay.
Because you said that this approach, biomarker-based diagnostics, right, kind of allows you to transcend clinical diagnosis. However, in this mixed group, I'm not sure if I would fully agree, because this mixed group, you only know they have both pathologies, but you don't know if one of them is more or less, and if one of them may be the driving pathology and the other. So it may be a Lewy body pathology with AD co-pathology. And if you have a DLB phenotype, that's most likely the case, but it may also be AD pathology with Lewy body pathology, and that may be a completely different thing, as we have seen in our brain paper, for example. So where we started with a clinical diagnosis of AD, and then a biomarker positive for AD, so it's definitely AD with Lewy body co-pathology, and these look virtually identical to the other, phenotypically identical to the other AD cases, with the exception that they showed a more aggressive disease and a faster cognitive decline, but they didn't show any evidence of mixed phenotype, no hallucinations, no posterior occipital hypermetabolism or whatever. So I think it's really important to keep in mind here what the clinical diagnosis and the composition is, because if you would just take dementia cases, what all of them, and use and define this mixed group, then most of them would probably be AD with Lewy body co-pathology, because since the AD is so much more common than DLB.
I think that's just a great comment. And what we are doing right now to trying to understand the contribution or the impact of these pathologies is to study the pure groups first, and get to know how does the fingerprint from a pure Lewy body, for example, look on the FDG PET or the FOBV PET, and then look at the pure Alzheimer group to understand what the individual contribution on the neurodegeneration may be when you see mixed groups. And I think this is a very clinically relevant way to tackle the situation, because if you in the clinic see a patient who is positive for both alpha-synuclein but also beta amyloid, and you consider starting an anti-amyloid treatment, you have to be sure that it's the amyloid that is causing the trouble and not the alpha-synuclein. And how can you do that? Well, maybe you can look at the pattern or the signature that you would expect there.
Yeah, that's because my question,
Yeah.
because you have to recruit these people, and I guess they are all dementia patients already.
Yes.
So, and you have to recruit them somehow. And so whether you recruit half of them, whether you recruit half of them with an AD phenotype and the other half with an DLB phenotype, or maybe uncertain cases, because that will affect a lot what you will find in this group, I think.
Yeah.
So if they are all recruited at your DLB consultancy, then you have a mix. Then you have Lewy body pathology with AD co-pathology.
Yeah.
But if you want to open it up to Alzheimer's, right?
Yeah.
And have
Yeah.
like a typical AD type dementia cases, then the results for this mixed group may be very different.
Yeah. I think that's a clear and strong point that you are making.
Extra question, final question. The FUV PET, do you do a dynamic protocol for the acquisition, or is just static?
When we do the whole body FUV PET is a dynamic protocol.
Because that would be a wonderful way of ensuring that this uptake is, of course, related with the cholinergic. It's specific. Have you analyzed the data?
Not the dynamic data, no, but my colleague he published a validation paper on the PET uptake in peripheral organs that I'm sure we can dive into to find the questions you may be looking for. Yeah.
I can offer myself to do it.
Yeah.
Okay. Any more questions for Niels now that we have him here? If not, you can also ask questions at our after work drink party at the Coronita Lofts. And yeah, thank you so much for being here, Niels, for a wonderful presentation.
Thank you for having me.